Good day, this is Professor Will, CBMD PhD, and today's Wednesday, November 24th, 2021. It is 11:06 and 55 seconds, a.m. Eastern Standard Time. Today's research, melanoma. So let's hop to it here. How moles change into melanoma. Date November 23rd, 2021. Source, Huntsman Cancer Institute. Melanoma researchers published a study that gives a new explanation of what causes moles to change into melanoma. And these findings pave the way for more research into how to reduce the risk of melanoma, delay development, and detect melanoma early. Moles and melanomas are both skin tumors that come from the same cell called melanocytes. The difference is that moles are usually harmless, while melanomas are cancerous and often deadly without treatment. Melanocytes are cells that give color to the skin to protect it from the sun's rays. Specific changes to the DNA sequence of melanocytes, called BRAF gene mutations, are found in over 75% of moles. The same change is also found in 50% of melanomas and is common in cancers like colon and lung. It was thought that melanocytes only have the BRAF V600E mutation. The cell stops dividing, resulting in a mole. When melanocytes have other mutations with BRAF V600E, they divide uncontrollably, turning into melanoma. This model is called oncogene-induced senescence. A number of studies have challenged this model in recent years. These studies have provided excellent data to suggest that the oncogene-induced senescence model does not explain mole formation, but what they have all lacked is an alternative explanation, which has remained elusive. The researchers took moles and melanomas donated by patients and used transcriptomic profiling and digital holographic cytometry. The transcriptomic profiling lets researchers determine molecular differences between moles and melanomas. Digital holographic cytometry helps researchers track changes in human cells. The researchers discovered a new molecular mechanism that explains how moles form, how melanomas form, and why moles sometimes become melanomas. The study shows that melanocytes that turn into melanoma do not need to have additional mutations, but are actually affected by environmental signaling. When cells receive signals from the environment in the skin around them that give them direction. Melanocytes express genes in different environments, telling them to either divide uncontrollably or stop dividing altogether. The origins of melanoma being dependent on environmental signals gives a new outlook in prevention and treatment. It also plays a role in trying to combat melanoma by preventing and targeting genetic mutations. Researchers might also be able to combat melanoma by changing the environment. And these findings create a foundation for researching potential melanoma biomarkers, allowing doctors to detect cancerous changes in the blood at earlier stages. The researchers are also interested in using these data to better understand potential topical agents to reduce the risk of melanoma, delay development, or stop reoccurrence, and to detect melanoma early. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.